Today our speaker for the CellTalk presentation will be Shweta Chakravarti. She's a scientist at Renix Life Sciences, and she's currently working on the project of digital medicine, exploring the clinical applications of wearable devices in monitoring the health status of cancer patients as compared to the healthy individuals. She has her master's in bioinformatics from Manipal Academy of Higher Education. So here is a budding researcher with about 1 year of academic experience. Over to you, Shweta.
Thank you for the introduction, Divya. So I'll share my screen. Is my screen visible to everyone?
Yes.
Okay, so hello everyone. So the topic which I'll be discussing is using drug nanocarrier as a drug delivery system in the treatment of glioma. And the research paper which I'll be presenting regarding the same topic, the title is Multifunctional Silica-Coated Mixed Polymeric Micelles for Integrin Targeted Therapy of Pediatric Patient-Derived Glioblastoma. So these are the authors of this research work, and it was published in the journal Materials Science and Engineering C, which has an impact factor of 7.328, and it was published in the year 2021. So if you have been following this webinar series, you must have come across this term glioma or glioblastoma multiforme quite a time. So I'll just give a brief introduction about this disease. So it is a very aggressive malignant brain tumor, which is usually found in the biggest part of the brain, that is cerebrum. And it originates from the glial cells of the nervous system. So they are mostly the part of central nervous system or the peripheral nervous system. And so these glial cells are the non-neuronal part of the nervous system, which just gives the support to the neurons. This disease has an incidence of 5 per every 100,000 people are affected with this disease. And it has been observed that 1.5 times more in men than women. Also, the disease has a very low survival rate, and like around 12 to 18 months of survival after the diagnosis, and only about 10% of the patients survive more than 5 years. So there are existing treatments for this disease, but they are not found to be much effective against the disease. So these are the surgery, radiotherapy, and chemotherapy. So these treatments are also used for other cancers, as we already know. So the treatments like surgery and radiotherapy, they are not very successful in completely removing the cancer cells. And chemotherapy also has its challenges and limitations, which we'll be further looking in this presentation. And this research can also have impact on other types of cancer, like breast cancer, leukemia. Since this drug delivery system can be used for other treatment of other types of cancer as well. So the main challenge which was encountered in this paper was the blood-brain barrier challenge. So this blood-brain barrier is the endothelial cells which lines the blood vessels. So it does not allow like 98% of the small drug molecules and 100% of the macromolecules to pass through it. So because of this reason, many drug molecules which are given for the treatment of the cancer are not able to reach the target cells. So it reduces the availability of the drug in the target region, and hence it cannot work against the disease. So when these cells are affected by tumor cells, that creates blood tumor barrier, and hence that name. But there could also be other reasons behind the failure of the treatment. That could be the unfavorable pharmacokinetics. So since pharmacokinetics of a drug is very important for its activity, like absorption, distribution, metabolism, and excretion. So the drug and also the drug and side effects could also be the other reason behind the ineffectiveness of the drug towards against the disease. So now I'll give the introduction about the polymeric micelles. So polymeric micelles is what was used in this research to as a drug nanocarrier So instead of directly injecting the drug, they came up with this nano carriers, which will act as a vehicle and carry the drug inside the target cells. So these drug nano carriers are found by assembling the polymers, which are amplifiers, and they have many advantages and properties which makes them a very good drug delivery system, like chemical flexibility and very small size, which let them pass through the cell membranes. And also they are easy to prepare, and they have a very high encapsulation capacity, which holds the drug intact. And also they have a good compatibility with the cells. So all these properties make them a good drug nano carriers. And the receptor mediated transcytosis is a very prevalent pathway which is usually used by these drug nano carriers to transfer the drug and transport the drug to and across the blood-brain barrier and to reach the target cells. So next we'll discuss about the integrins and PTV. So here in this work, the integrins were the target, which was really targeted for the treatment, and PTV is the drug. So integrins are the transmembrane receptors which are found on the cell membrane, and they have many different functions, like they have function in interaction between cell to cell and extracellular matrix. So these interactions also lead to different signaling pathways, which further becomes very much like involved in the cancer growth, like cell division, proliferation, which leads to cancer. So these are the integrins, and these are also overly expressed in the BBB, that is the blood-brain barrier. And in this research, they have also used something called cyclic short peptides along with the drug nanocarrier. So these peptides are found to have a very high affinity towards these integrins, and due to that, it makes the drug nanocarrier to pass through to reach the targets easier. And the drug, that is pitavastatin, which was used here to be carried by the drug nanocarrier, that it is an anti-cholestromic agent. So it is known from the literature previously that it has anti-GBM or anti-cancer activity. Hence, this drug was used. So the overall objective of this research paper was to evaluate this drug nanocarrier as a drug delivery system using two polymers which it was prepared, and also to assess its compatibility with the cells and how well the cells are able to take these polymeric micelles, that is the drug nanocarrier. And it was also used to evaluate its anti-proliferative and apoptotic activity to show that how well it is effective against the disease glioma. And for that, they had taken the cell line from the pediatric patients, GBM patients. So, it had a very vast methodology, which has a complete wet lab experiment, so using different chemicals, reagents, and apparatus. So I'll be briefly going through each step, telling its significance. So first 3 steps was basically involved in the preparation of the drug nanocarrier, which was using two polymers. So the first polymer was also conjugated with the cyclic peptide, which and I mentioned about its importance, and we'll further see why it was useful. And another polymer was conjugated with this green fluorescence labeling, that is fluorescence isothiocyanate. And further, these two were mixed to form the polymeric micelles, and it was also coated with silica shell, which was very useful to maintain its stability. And then the drug PTV was loaded and encapsulated inside this polymeric micelle. Further, the polymeric micelle was characterized to understand its structure, its shape and size, and also its storage stability was also understood. Further, it was further evaluated to show how good this is as a drug nano delivery system. So for that, they've analyzed how controlled way in this it releases the drug. Since we know the pharmacokinetics of the drug is very important, the drug needs to be released in a very controlled manner. So In the next steps, the cell compatibility and cell uptake was analyzed, and for that they had taken cells. These are three different types of BBB cells, cell lines which they had taken, and they did the cell culture techniques, performed assays like MTT assay, and also the same technique was used, MTT assay, to analyze the anti-proliferative and apoptotic activity of this drug nanocarrier. And for that, this cell line was, glioma cell line was taken. This was derived from the pediatric patients. And finally, they also quantified the mRNA levels in these genes, which are known to be associated with glioma and apoptosis. So these the mRNA levels in these genes were further quantified. So now coming to the results. So from all the stability analysis and the preparation of the polymeric micelles, they found that the entire structure of polymeric micelles was quite stable. And the silica shell also helped in maintaining the stability and didn't allow it to disassemble in any case of extreme dilute conditions. So it was quite stable and remained intact. So this is the chemical structure of the drug. And these they are showing the two polymers, which was conjugated here with the peptide, cyclic peptide, and here with the green labeling, fluorescence labeling. So it was further prepared to and inside this and to form a micellar structure. And inside this PTV drug, that is the hydrophobic drug, was encapsulated. So this forms a, as I mentioned, it is an amphiphile structure. So it has the outer regions are hydrophiles, and inside it's the hydrophobic region. So these structure makes it easier to cross the cell membrane. So next is the result of the drug release, and the in vitro drug release, which was done to understand how, in what manner the drug is released inside the blood. So these are three different samples when only drug was injected, and this is when it was injected with the polymeric micelles, that is a drug nanocarrier. In this case, the silica shell coating was done, and in this case, there is no silica shell coating, as you can see from here, the labeling. So we can see in case when only drug was injected, a lot amount of drug is injected in a very less time, and this can also lead to off targeting, since as soon as the drug is injected, if a lot of drug gets released inside the systemic circulation, it can lead to the off target. So we have to it needs to make sure that a right amount of drug reaches the right region. So we can see compared to silica shell coating and no silica shell coating, the silica shell coating is ensuring a better release and better release of the drug in a much controlled manner as the time is passing. So we can see this advantage of silica shell coating in this polymeric micelles. The next result is showing the cell compatibility and uptake analysis. So it was done on the BBB cells. So these are the three plots for three different BBB cells which was used, blood-brain barrier cells. And we can see from the plots that most of the different samples which are used with like using polymeric micelles, as compared to control, the cell viability was very good, and in most of the cases it was above 80. So which is good that these BBB cells were accepted by the PM, like polymeric micelles were accepted by these BBB cells, and it was internalized. And these figures are also confirms the result. We can see the green fluorescence, which was conjugated inside the polymeric micelles, as I mentioned, is inside these BBB cells. So we can tell that as the challenge was that these drugs are not able to cross these BBB cells. So we can say that the BBB cells were able to accept these polymeric micelles due to this presence of green color here in these figures, and overcome that challenge. So, so next result, they're showing the anti-proliferation and apoptosis activity inside the glioma cells. And this result, for this result, that they performed it on the glioma cell line. And here the plot, they're showing the cell proliferation concentration. So when they, in these last three, where there is drug is used with the polymeric micelle and the cyclic peptide and also the silica shell coating, we can see the cell proliferation has reduced very significantly. So we can tell due to this reduction in the cell proliferation, we can say that it is showing the anti-cancer activity. And in the next panel of these results, they are trying to show that this PM, so this green color, they're indicating that the polymeric micelles have reached the target cells also correctly due to this presence of green color. And these are the GBM cells. So, and these blue color are the nuclei. And they're also trying to show that the DNA was more fragmented in this case, and the size of the nuclei also reduced as compared to control, so which happens during apoptosis. And the next panel of results confirms that the apoptotic concentration of the nuclei has increased when they have used PTV loaded silica shell coated and polymeric micelles, which is also modified with the surface modified with the peptide, and it has the highest concentration of apoptotic nuclei. So it tells that these polymeric micelles are showing the apoptosis, and because of these, the apoptosis is taking place. And here the another result, this is showing the cell structure, and when this drug nanocarrier along with the drug was used, the roundness of the shell was lost in this case. They visualized under the microscope, and it is showing some kind of apoptotic bleb, it is known as, and it happens when the apoptosis takes place because of the disruption of the cell membrane. So this is the last result where they're showing the mRNA level. So since these genes are associated with the molecular changes which takes place during glioma, and they also induces the inhibitors of apoptosis. So we observed that the mRNA levels have significantly reduced when the drug was given. There's a significant reduction in the mRNA levels, which tells that which tells us apoptosis can take place because these are associated with apoptosis, and there's a reduced reduction and reduction in the gene expression of these genes. So overall, all these previous results suggest that these silica shell coated and surface modified peptide polymeric micelles are easily internalized by the blood-brain barriers cells, and they also increases the availability of these drug inside the cells, inside the cancer cells, since they are compatible with the cells. And due to the high affinity of this peptide for the integrins, which are the targets, it increased its availability inside the cells, and also increased its anti-cancer property. So the complete impact of this research was, since as I mentioned, the very big challenge of blood-brain barrier was could be overcome in this case using drug nanocarrier instead of only using the drug for the treatment of cancer. And due to its numerous properties of drug nanocarrier, like it makes sure the drug is released in a controlled manner, and it has a good drug encapsulation property. It makes a very good drug delivery system. Along with that, these peptides, the cyclic peptides, which was modified using this peptide, they also increases the nanoparticle uptake inside the cells, and also increases its apoptotic activity and the anti-cancer activity. So that was about this research paper. Thank you so much for your attention and patience for listening.
Thank you, Shweta, for this enlightening and an informative presentation. So now we'll move on to the questions from the audience. Presently, I have a question from Dr. Vijay. He wants to know, how did they infer specific internalization by only BBB cells and not by any other cells?
How did they only BBB cells?
Yeah.
Okay, so as I mentioned that because of the main challenge which they were focusing in this research paper was that these BBB cells, that is blood-brain barrier, they don't allow the drugs to pass through them. So that's why they wanted to focus on this BBB cells, and that is the reason they chose the cell line to show, and because of the presence of those green color, it was made sure that yes, they were internalized by the BBB cells and accepted.
Thank you, Shweta. Any other questions from the audience? We'll be ending this session. Thanks one and all for your patient hearing. And also, once again, thank you, Shweta.
Yeah, thank you so much, everyone.